I will address the issue of how we can make headway into improving cancer therapy for GBM and cancers in general. And the position that I've and everyone can take is the efforts that we have expended at finding therapies have not been very successful, especially for something like GBM, partly because the tumor is complex, it's heterogeneous, it's difficult to access, and we understand very little. And the lack of knowledge has really been a handicap in finding the therapies that would work for a given patient, given the patient's characteristics or the tumor characteristics. In an ideal world, what we would want to do is look at the tumor profiles from a multi-omic perspective. So look at the mutations, the epigenetic landscape, the gene expression patterns, the cellular heterogeneity, and so forth, and figure out how every patient is unique and map that patient to a disease-perturbed network that is very specific in terms of the dysfunction underlying their disease, and use that knowledge to then find the therapies that are most likely going to work with that patient. So that is the right arm of this schematic here, where you take a cohort of patients, and from those patients, you then identify signatures that can stratify the patients and map each patient into some causal mechanistic flow of information in a network, and then use that information to find therapies that can target specifically elements of those networks. And the other arm I will not speak about today, which is that even within a given patient's tumor, the amount of heterogeneity is so enormous that the therapy that you might use may work against some or a lot of the cells, but not necessarily all of the tumor cells. And as a result, because of tolerance and resistance phenomena, you have recurrence. And to be able to have therapies that are long-lasting, progression-free, you need to get rid of the entire tumor cell population. And to be able to do that, you need to have a similar approach where you understand the heterogeneity of the tumor, find what the dysfunction is in all the different cell types that make up the tumor, and then design your therapies to get at all of those cell types. How would one go about doing this? So let's imagine for a moment that you have a cohort of patients, and it's a large cohort, and within the cohort are subgroups that share similar dysfunction and mechanistically similar causes for the disease. The challenge is to now mine the multi-omics data, so to now stratify those patients in a way that can subgroup them based on the mechanistic dysfunction, and ideally even rank order them based on the progression stage or subtype of disease. And the challenge here is that when you have a dysfunction in a network, you have the consequences propagate throughout the network. So you may have a dysfunction in one gene, but the genes that the dysfunctional gene interacts with are also perturbed, and so are their downstream products. And so when you make measurements of multi-omics profiles, you actually are not measuring just the dysfunction per se, but all the primary, secondary, and tertiary consequences, and you end up with these very large data sets. And technologies are only getting more powerful, so you're able to measure even larger amounts of secondary, tertiary changes at different levels of information. The approach one takes, and the buzzword is big data and machine learning. And in simple terms, what that means is when you have a very large amount of data, you find features that are correlated. And the assumption is when you have a change in one feature that correlates with a change in another feature, then it implies some kind of functional association. And this is extremely powerful and is one of the major ways that big data are mined. Of course, there's a lot of complexity in how you set up your machine learning algorithms. But at the heart of it, you're finding correlations. Now, we are dealing with cryptic information. You can have some code for epigenetic marks. You may have some code for gene function, for a mutation. We don't know what each of those do, but let's take data sets where we do know what the different features mean and see how powerful this approach can be, right? So just bear with me for a minute here. So these are data sets from USDA and CDC that tell us that if you do machine learning on those data, people who drowned after falling out of a fishing boat correlate with marriage rate in Kentucky. The age of Miss America correlates with murders by steam, hot vapors, and hot objects. And look at the profiles, they're exceptionally correlated. And the per capita cheese consumption correlates with number of people who died by becoming entangled in their bed sheets. And it's funny because we know what each of the features that we've correlated mean, but when you look at multiomics profiles from tumor tissues from patients, we almost always don't know what these proteins do, what these mutations do. And when we draw this graph of mutation associated with gene function, that implies that there is some causal mechanistic association, but it could be just something that is completely coincidental. So how do you prevent getting caught up with such meaningless correlations? How do you go from correlation to causation and ultimately mechanism? If you want to perturb behavior, you need to know mechanism. Causation alone is not sufficient. And the way you would do that is by this approach of reverse engineering, but within a framework that takes into account principles of regulation and biological function and processes and so on. So the idea is you can treat patient cohorts or a cell or any complex system as a black box that has some components, and then you perturb that. And in case of patient cohorts, there are already a perturbed set of individuals. And then you look at the responses, which should be the multiomics profiles. And then from that, you reverse engineer the topology of interactions. Now, this is the key part. How do you reverse engineer those interactions? You have to put constraints that come from known biological principles in terms of how gene regulation happens, how epigenetic markers are changed by enzymes, and what the consequences are. So you can impose meaningful constraints based on prior knowledge. And you can do cross-validation across data sets, independent data sets. And if you do all, and I'll show you how to do that, then you can reverse engineer topology of interactions that is true to the data and true to the principles that you have constrained the discovery with. And then the hope is to have that network model be predictive so it can explain all the perturbed responses that you observed, and it can predict how you can intervene and reverse some of those perturbed states, disease states perhaps. So this is an example of a multi-clustering algorithm. Without going into too much details, this algorithm will be looking at gene expression profiles in the top left panel, where those plots indicate gene expression patterns, and the different colored lines indicate expression patterns for different genes. And then you have a vertical line there, which separates some patients on the left and some patients on the right, and that is indicative of which patients have correlated and high covariance of changes in the genes versus which patients don't, and they're excluded. And then we have a network component, which is what I was describing. So you can take information regarding protein interactions, protein DNA interactions from literature, from other predictions that you might have, and then constrain the search space, where you can say, given a set of genes in a cluster, you should prioritize these other genes because I think they are somehow functionally associated for these reasons, right? And when you have enough genes that get grouped into a cluster that have correlated expression profile, you can start asking other questions. Is there evidence that they're co-regulated? Is there a mechanism for why these genes are being grouped together? Can I find in their promoters or in their upstream or downstream regions or their transcript untranslated regions evidence for molecules that might be binding to mediate their similar patterns of change? And that's an independent source of information. So you have a scoring function that takes into account multiple pieces of evidence before you can conclude that these genes are co-regulated. So this is one way of preventing coincidental correlations that are meaningless, because it's unlikely that you have patterns that are supported by different pieces of evidence, many of which are based in mechanistic principles. Right? So here's the algorithm in play. It is scanning the patients, scanning the genes, and then de novo discovering sequence signatures, and you can look for mutation patterns, epigenetic profiles, and so on, and find for consistency across those different pieces of evidence. Doesn't mean every piece of evidence has to be supportive, but you need a critical number of evidences that need to be there to make it significant. And once you find this module, then you essentially found a signature that is stratifying patients potentially based on some similar expression patterns. And you can do this in parallel and find thousands of signatures from very large data sets, from thousands of patients at a time or more. And once you found this module, then you have to infer what was the underlying cause for the dysfunction. And there's a number of ways you can do that, and I'll get to that in a minute. But ultimately, what you end up with are predictions that kind of look like this, where you have some environmental factors, maybe you may have some regulatory factors that you can decipher to be associated with driving the change of gene expression within each of these signatures. And then you can use that model to make predictions. And what we've shown is that you can make these predictions even for patients that are outside of the cohort from which you derive the models. That's a key point, is you can overfit models by taking large data sets, and your model will perform beautifully on that data set. But the key thing here is when a new patient comes in who is not part of the model construction, does that patient benefit? So your model needs to be able to have that predictive power. And we've shown over the years across multiple systems, not just for cancers, but in diatoms and tuberculosis and in microbial communities, that you can apply this approach and build very predictive models. And you can use that to then make discoveries. So now let me spend a little bit of time in describing specifically for human networks how we get at the mechanistic connections, the wiring diagram within these networks. So let's say you have a set of genes and you want to know if there's a common regulator responsible for controlling those genes. Now, one possible way that could happen is by a transcription factor, a protein that is binding to the DNA region, the control region or promoter of those genes, some or all of those genes, to mediate coordinated upregulation or downregulation. Okay. The second way it could happen is if there's a small microRNA that goes and binds to their 3' untranslated regions and destabilizes the transcripts. And there's several other ways, epigenetic marks included. Let's take the first one. So here's the challenge. When you look at the human transcription factor set, there's thousands of transcription factors. And typically each transcription factor recognizes a short 6 to 8 nucleotide sequence. And if you look at the matches to that sequence across the genome of 6 billion base pairs, the frequency of distribution of those matches is close to random. And so you need to figure out which of those matches are actually functional. And one way to do that, and I'll show you with this example, is with the USF1 protein, is it recognizes a short sequence, CAC, GTG, in the 3 locations I'm showing you in this example where there are matches to this recognition sequence. So potentially this protein could bind in any of those 3 locations. Then we layer in a second source of information, which is chromatin accessibility. So this is a DNA's hypersensitivity mapping, which essentially maps where in the genome was the chromosome open and accessible to the protein. So of the 3 sites, we find that only 2 were accessible, the 2 peaks that you see. Now, among those 2 accessible sites, we ask, is there evidence across all the 200 cell types that were profiled if there was a protein actually bound to that location? So we zoom in and we look at whether there was a protein protecting that DNA from the enzyme cutting, which is used to map accessibility. That's called a footprint. So you find that of the 2, only one of the sites, the one in the middle, has a footprint. So through this layered search process, we find that the USF1 is potentially active only in one location across at least one of the 200 cell types in the human system. And I'll skip this one here. So that is how we map the wiring diagram on the protein side. And on the microRNA side, what we can do is we can ask, is there a microRNA that is controlling a set of genes that are in your signature that you've discovered? And this you can do in one of 3 major ways. This is one that we invented, which is to find whether the genes that are in that signature share a short conserved region that could be a potential microRNA binding site. And these are, again, 6 to 8 nucleotides long. Then we take the frequency of nucleotides at each position along the 6 to 8 word that you conserved word that you identified, and convert that to a hidden Markov model. So essentially probabilities of finding that string. And then we can go into a database called MIRBase, which has all the known sequences of microRNAs that recognize these short words. And then from that, we can identify what microRNA must be regulating. So through this reverse engineering process, I just wanted to give you 2 examples to give you some appreciation for the depth of analysis that is necessary to really dig deep and find mechanisms and not just stop at finding correlations. Okay? I'll skip this as well. So this is the platform. There's dozens of such steps that we take to go from the multi-omics profiles of patients to this causal mechanistic flow of information to predict how mutations, somatic mutations in the tumors have perturbed specific regulatory pathways and their downstream targets to perturb hallmarks of cancer. So in glioblastoma, we took about 420 odd patients, and from them we discovered this flow of information where we found 34 mutations in genes, 68 mutations in pathways that were perturbing 74 transcription factors and 39 microRNAs. And the perturbation of those regulatory elements resulted in altered expression of about 5,000 genes that were in 500 signatures and that perturbed 9 hallmarks of cancer. So how do we use, so this is just what I told you about how that network was built, so I won't belabor that point, but let's look at the features of this network and how that could be potentially used in the clinical setting. So here's a signature that was derived from the patient cohort where you have about 20 odd patients that are subgrouped based on 16 genes that have coherent expression patterns across those about 420 patients and 16 genes. And the patients are rank out. So every point here is a patient. So remember, this is cross-sectional data. And the biggest question we had was, can you get pseudotemporal trajectories of disease progression or subtypes from cross-sectional data? And this essentially suggests that potentially yes. So you can rank order the patients and then mark them based on subtypes that have been described. And I think Eric referred to those subtypes earlier. And you find that in each of the subgroups, and as you go across the trajectory, you have different clinical subtypes that are enriched. And it becomes more mesenchymal if you go to the extreme right, and it is predominantly normal tissues or proneural types on the left of this. Now we can ask, is this trajectory independently predicting some clinical outcome? So you can take patients from different quintiles, different groups here, and ask if patients from the lower quintile and patients from the upper quintile had different outcomes. In this particular cohort, we had survival information, and we could do Kaplan-Meier plots and show that, yes, patients in the upper quintile, when genes were highly expressed, survived for a shorter period of time than patients who were in the lower quintile. And if you have drug responsiveness information, which we've done from multiple myeloma, we've shown that using pre-treatment profiles, we can predict which patients will have progression-free survival, and the Cox proportional hazards ratios that we get based on that segregation is unheard of. It's in the tens. So this type of approach really has the potential of making predictions independent, because we did not train the algorithm on survival information. That was used post-hoc. It's a really important point. And for each of these signatures, we've derived this type of mechanistic flow of information, causal mechanistic flow of information. So for every cancer phenotype, we can find out all the signatures that are associated through the genes that are marked with that function, and then identify what mutation has perturbed that cancer phenotype. Okay. Now this allows us to construct this really big complex network model, but for each edge here, where you have mutations shown as chevrons, the triangles shown as the transcription factors shown as triangles, and the microRNAs as diamonds, and then you have signatures as little squares down there, and the cancer phenotype. So we have this complex map of how mutations are acting in different combinations to perturb different pathways and having complex effects on cancer phenotypes. And we can now mine this for testable hypotheses. This is one of them, where we looked at the distribution of mutations that were causally and mechanistically predicted to affect lymphocyte infiltration. So we found that mutations in NF1 or PIK3CA were predicted to causally increase the expression of this protein called IRF1 that was predicted through that wiring diagram I described with the ENCODE, the profiling information. It was predicted to regulate about 27 genes that were involved in tumor lymphocyte infiltration. And we can look at each of these edges and then further look for evidence in the data set that you used to build the model and independent data sets that do patients who have these mutations have increased expression of IRF1. And that's what is shown on the top left there. We see that mutated tumors do have increased expression of IRF1. We can ask if those tumors that have increased IRF1 expression have increased expression of genes in the signature, and you can confirm that, and so on. So you can confirm each piece of evidence, and then ultimately you can ask, well, is there evidence at the phenotypic level that tumors that have these mutations do have perturbed antigen presentation and processing and presentation? So we took the tumor H&E stains, and then we looked at evidence for increased lymphocyte infiltration across tumors that had mutations or increased IRF1 or increased signature expression. And turns out there is a correlation between the perturbed pathway and the immune phenotype. Now, once you have this type of information, you can start perturbing the network. If this is truly a causal mechanistic model, then we should be able to perturb the cancer phenotype by blocking the causal flow of information. You can do it with drugs, you can do it with genetic interventions, or small molecules targeting microRNAs or transcription factors. And because we know the topology, in many cases we can predict synergy. So you can have drugs that are attacking a transcription factor or a protein product, and the transcript of the same protein product, you would get synergy. Here's one example where this is work we did with Mike Jensen, where as part of the City of Hope trial, they had observed that patients who were treated with CAR T cells against IL-13 receptor alpha 2 came back, and there was a hypothesis that this might be because of the under expression of this receptor. And so he asked us, can you find in your network evidence for some regulatory network around this receptor that we can perturb to increase the expression of the receptor? So you can think of combination therapy where you pre-treat with a drug that makes all the tumor cells express the receptors, then you treat with CAR T cells. And again, we confirmed evidence that this flow of information was correct. What the network said was when you have mutations in interferon gamma pathway, there's a transcription factor, which I can't disclose right now, that controls a set of genes that are involved in evading immune detection. And so we predicted that if you inhibit the transcription factor, you should increase the expression of the receptor. We found a small molecule that targets the transcription factor and showed that at very low doses of that small molecule, we could increase the expression of the receptor. And this is about 100-fold lower than the concentration that would start killing the cell. So this would be like an adjuvant. And this is the way you could use this network model. So when a new patient comes in, there's multiple ways you can map the patient into the network, through their mutations, through their gene expression patterns onto signatures, epigenetic marks, and so on. And once you identify the patient into a subgroup, you can predict how the patient is going to do based on what the information you have on those patients regarding drug responsiveness, clinical outcome, what have you. But you also know the perturbed network underlying the signature to which the patient is mapped, and based on that, you can predict what molecules are likely to work on that particular patient. And this is where you get to the N-of-1 type trials, where you can then take patient-derived cell lines and glioma stem-like cells, the kind of work that Charles is doing, and set up these screens to find out whether you do have synergistic effects in combinations of drugs in the patient-derived cell lines. Now, this is where I think we often fail, is there might be a drug that is really effective, but effective only in a small subgroup with certain characteristics. We don't know what those are. So when you try to replicate the studies across different trials, we fail to do that. But if we know based on some preliminary analysis or this type of a model, you can then recruit patients specifically based on some hypothesis regarding what their network looks like. And in that way, you can increase the likelihood that your trial is going to be successful. And in this case, we're showing for, and this is with a very toxic drug, romidepsin, that is known to inhibit proliferation. And the question here we were asking was, can we find a second molecule that can help us lower the dosage of romidepsin but recover its activity through some synergistic interaction? And we found a small microRNA that we could target and accomplish that. So essentially what I showed you was we went from this large cohort of 420 patients and reverse engineered from those patients these network models onto which we are now able to map patients and subgroups of patients and then identify the underlying dysfunction to then target those perturbed networks with rational interventions. We're doing something very similar. I don't have time to speak about at the single cell level, where we're taking tumors from patients and looking at the complexity of the different cell types and finding networks within individual cells. And you can imagine the same concept, but now we're looking at combinations of drugs targeting different subpopulations of tumor cells. So this work is published, and I can send you more information. I don't know if it's entirely, it's in Cell Systems. The first author is Plaisir. It was published a couple of years ago. And this, as I mentioned earlier, this method is effective in finding combinations that are in clinical trials, but we've not done it for cancers. We're in the process of doing that. But for tuberculosis, a combination we predicted is in clinical trial, where we predicted that bedaquiline, which was FDA approved, has delayed killing effect on the pathogen because the pathogen, when it sees the drug, it elicits this tolerance response. And we figured out what the mechanism for that tolerance network was and found a molecule that disrupted that tolerance network and showed that when you combine those two drugs, you get a synergistic effect and it improves killing. So in summary, you can find patient-specific perturbed networks, and you can do it from multi-omics profile, but there needs to be some care in terms of going from correlations to causation to mechanism, and that is key to avoid ridiculous correlations, wasting your time, and to be able to rationally intervene with therapies. And because we know the network architecture, there is potential of finding synergistic combination. The space of combinations becomes exponentially larger and difficult to screen with the few cells that you can derive from the patient tumors. So you would like to prioritize drug combinations, and having a network that does that for you is very helpful and does so in a personalized fashion. And so the vision then would be to go from these network models to go from patient cohort multi-omics data to network models to N-of-1 trials. So I'll stop with that, and have to acknowledge Chris, who's in the middle, who's now a professor at Arizona State University. He was a powerhouse in my lab who did a lot of the cancer networks. Eliza, who's next to him, is the lead person who did the tuberculosis work. And there's a whole bunch of other people. Matt on the lower left there is doing multiple myeloma work in a partnership with Celgene. And the results are looking spectacular over there. And so I'll stop there. I think 50 seconds left.